So I want to talk to you about the MS Stem Biobank today, and I think a good place to start if we're going to talk about this is initially what are stem cells. We're also going to cover where do the stem cells come from, how can we use stem cells in MS research, or how are we using them in MS research, and how does the MS Stem Biobank operate. And so to start with the first question, here we go. What are stem cells? So stem cells are immature cells. They can divide and make new cells, and they can do this indefinitely. So you could keep the cells growing for years. They also have the ability to make multiple different cell types from the body. So you could have one stem cell and you could push it and it could make muscle cells, or you could push it a different direction and it could make fat cells. Or one of the things I like to do is push them to make brain cells. So in terms of our own body and our own development, the ultimate stem cell is the fertilised egg. From the point of conception, that cell has the ability and does make every single cell of every single tissue in our body as we grow. We then end up in a situation where the different organs become a bit more specialised. And so within the muscle, you'll have stem cells that can replenish the muscle. Within the fat, there'll be other stem cells that can make more fat cells. And so as an adult, we don't have a single cell in our body that has that ability that the fertilised egg had, which is to make anything and everything, because our tissues do become more specialised as we grow. But that doesn't mean we can't end up with stem cells again. So even though true stem cells do not exist in our body, we can make them in a laboratory. And this is because of key discoveries that were made by two people that were co-awarded the Nobel Prize in 2012. So John Gurdon discovered that a mature cell has exactly the same DNA as an immature cell. That means it's still got all of the information inside it that it had when it was an immature cell. And if we could just tap into that, it could sort of rediscover that ability again. And then Shinya Yamanaka discovered how to actually do that, how to reprogram mature cells to almost push them backward in time so they can remember their origins and have that ability. And that is what we can now do and what we have been doing as part of MS STEM. We've been asking a number of people in the MS community, both with and without MS, to donate blood. And then we take those blood cells, which are mature cells doing their normal function, circulating through our body, and we push them back in time, we reprogram them. And this is what we get. We get cells that are called induced pluripotent stem cells. So they have been induced to be stem cells in the lab. And pluripotent is just a science word for saying can make every cell type of the body. And so effectively, that is the definition of a stem cell. We then have them in a dish where they can divide and make lots and lots of new cells, and we can work with these in an ongoing way in research to try and make cells like brain cells, which gives us a way of studying brain cells from people with multiple sclerosis without us having to, really inappropriately ask for brain samples. And so we've been going to the community and we've asked about 100 people with and without MS to make this blood donation. And when we take the blood cells, they have the same DNA as all of the other cells in your body. And we have then reprogrammed them to stem cells, which also have that same DNA fingerprint, which means that these cells have the genetics of the person with MS. So even when we then take them and make them into brain cells, they've still got that DNA fingerprint that makes them part of you. So they can be expanded in a culture dish. We can freeze them. We can store them for a really long time. We can use them here for our MS research, or we could send them to Anne in Canberra and she could use them for her MS research. And because of the fact that they have the DNA, that actually allows us to also study how a person's genes ultimately make a brain cell behave the way it does, or how it might behave differently in a person with MS than it might behave in a person without MS. And one of the things that I really want to show you today is actually some of the pictures of the cells that we've been making, so that we can try and figure out, like we've been collecting images of these cells and we've been taking measurements from them to try and find out if there is something intrinsically different about the brain cells from people with that develop multiple sclerosis. And the other thing then that we can do with that, if that is true, is use these cells for drug screening purposes to try and bring new brain repair treatments to the MS community. So when we take a blood sample, it then can take a number of months to generate each of these stem cell lines. The blood cells start out as the single cell on the day 0 little square, and I'm sorry, I thought the screens were bigger here than they actually are. Once we reprogram them, they kind of swell and they get a bit bigger, and then after that they stick down and they adhere. So by day 3, they're starting to stick to the bottom of the dish, and then they're starting to grow to form these colonies. So by the time we get more towards sort of day 10, we end up with these little clones of cells, and these are the stem cells. But it's really hard sometimes to imagine what this is like if we just look at it in static pictures. So I asked Ariane, who is the manager of our MS Stem Biobank, to go into the lab and try and make me some videos of what this looks like in real life. And here we go. So this first video are some of the very, very early clones. And what you might be able to see from either the back monitors or the front monitors is that there's a clone in the top where some cells come out of it, they move around quite a lot, and then they go back into the clone. And so it's not a static structure. We sometimes think of, we put a cell in a dish, but we don't realise how much these cells are actually moving around. And while it might be creepy, this is also what happens with cells in the brain. They actually do move around quite a bit. And so we end up with a smaller number of cells becoming a larger number of cells. And I think this is what happens a lot in the second video, which really sort of emphasises how much over time these clones are growing. And as it's spreading, that's the generation of new cells that's actually allowing that structure to get bigger and bigger and bigger and bigger. And so this is how stem cells can renew themselves and generate new stem cells over time. Once they get really big, we then cut them into little pieces, and we take a small amount of that clone, put it in another dish, and it will keep growing. So we can keep this going indefinitely. In terms of the brain cells we make from them, these are some oligodendrocytes. So a large number of people in this room will have heard of oligodendrocytes because they get attacked by MS. And so we thought, well, is there something that could be different about the oligodendrocytes themselves in people with MS? It actually turns out that for people with relapsing remitting MS, the oligodendrocytes in a person that does and doesn't get MS are remarkably the same. So they do seem to be more like victims of the disease, really, rather than cells that are causing the disease. However, in people that get primary progressive MS, the oligodendrocytes do actually seem to be less resilient and maybe less able to repair as well. And so that is something that then we can try and target with future therapies. But other cells that we've been making the stem cells into are these cells, which are pericytes. They also move around a heck of a lot in a dish. These are cells that are part of brain blood vessels. And they actually squeeze around the capillaries and regulate how much blood flow goes to different parts of the brain. Work by our PhD student Alastair has actually also shown that in people with MS, pericytes are a little bit bigger than they are in people without MS. And so we're trying to figure out if that affects the blood-brain barrier. So maybe that's part of why the immune cells actually get into the brain. I don't have a definite answer for that yet, but that is work that we're continuing with some ongoing NHMRC funding. And the other cell type from the brain that we've been looking at quite closely are cells that are called astrocytes. And this is what they look like when we make them out of stem cells in a dish. And again, work from Alastair's PhD has shown that in people, when the cells are generated from people with MS, these astrocytes, which also affect the ability of cells to get in and out of the brain, like in terms of the immune cells, are actually more prone to respond to inflammation. So it's like they've been primed to respond more strongly. And so our research is really telling us that actually the DNA or the genes of people with MS are contributing to how the brain cells are behaving, and that should actually be a primary focus of the way we try and approach brain repair treatments going forward. That this is a potentially new target for future treatments. And here we go. So we're now custodians of 162 stem cell lines. This is what makes up the MS Stem Biobank. And so this is just a breakdown. We've got stem cells from people that have relapsing remitting MS at the time of blood collection, or secondary progressive or primary progressive MS, or did not have MS. So we've got cell lines from a variety of people. And we can now make some of these stem cell lines available to other researchers to also try and expedite how quickly we can get towards those brain repair treatments. Other researchers can access the website, which Tash and Rachel talked about earlier, and they can contact us and say that they want to collaborate. We also, as people publish more research using these stem cells, will make those publications available on the website. But we do not give the stem cells out to other people without due consideration. So we have a governance committee. It has 7 members, of which I am one. Ariane, who's a number of you will have met because she's in the room, is another, and Natasha Stevens. But we've also got Megan Menzies, who is a stem cell research ethics expert, and Joe Dickinson, who is an expert on biobanking and human research ethics. And we've got 2 members of the MS community. We put out a call for expressions of interest, and Charlene Brown and Jane England, I'm very happy to say, are now part of our governance committee. So what that means is that when we have researchers approach us and say they're interested in accessing this stem cell resource, we will look as a committee at the application and make sure that they've got appropriate research ethics approval, that the sort of research that they're trying to do is something that would be valued by the MS community, and then from there we can approve the distribution of those cells. And so that's all I wanted to tell you about today. Thank you.